Welcome to Chapter 7, Part 2. We are going to start moving into those actual pathways by which energy is going to be made within the cell. So, this is where we ended with Part 1 with oxidation reduction reactions, or known as redox reactions. Now, the important thing to remember about oxidation versus reduction is we are talking about either receiving or losing an electron. That is the most important difference. Now, with oxidation, what's going to happen is the molecule, and in our image up here it is molecule A, is going to have an electron that it's going to lose. So this oxidation is happening where molecule A is losing an electron. What's really important here is that in this process of losing the electron, that means that that molecule is losing energy as well. With reduction, that's going to be our molecule B. It is receiving the electron, so it is gaining an electron, and that is the reduction process. Now, anytime you have this happening, you are going to have both oxidation and reduction occurring. So it is a redox reaction. Both of them are happening simultaneously. It's the same time. So as molecule A gives up its electron to molecule B, molecule B is going to gain energy in the form of that electron. We'll talk about what that means here in a bit. By getting that electron, it is gaining energy. Redox reactions are going to be pivotal in moving energy around. And two of the most important redox reactions are going to come from our electron carriers that we're going to make through a couple steps that we're going to talk about soon. Those electron carriers are NADH and FADH2. Now, once these guys are made, we are going to talk about the process of turning ADP into ATP, but that's going to come at the very, very end of the pathway that we're going to discuss. Just know for now that these two molecules that are produced allow ATP to be made because they carry electrons. That is going to be a pivotal point in this pathway that we're going to discuss. Now, this is another example of a redox reaction, not dissimilar to what we looked at on the first slide in this lecture. Now, what's happening is we have an NAD plus molecule that is going to gain electrons from some organic molecule. Doesn't really matter what it is, it's just an organic molecule that is holding electrons. This NAD plus is going to get those electrons in an oxidation reaction. So that organic molecule is losing the electrons, and in a reduction reaction, the NAD plus molecule is going to gain the electrons. Now, if it helps, there are little acronyms you can use to remember oxidation versus reduction. There is Leo goes GR, which I think is kind of cute. It's a good one. For LEO, I want you to make sure that you know that loses electron equals oxidation, so LEO, and then GR is going to be gains electron equals reduction. Make sure you know that for Leo, loses electron oxidation, that this also means that that molecule is losing energy, while in GR, gains electron is reduction. That is going to be where the molecule gains energy in the form of that electron. You can also remember with oil rig, and what this stands for is oxidation is loss, where RIG means reduction is gains. So however you want to remember oxidation reduction reactions and what's happening with those specific terms, I love it. Fantastic. Just remember one of them. You don't have to remember both. As long as one helps you remember it, that's all I care about. Now back to those electron carriers. Remember that is the NADH and FADH2. This means that they are going to hold electrons or energy for a short period of time. This is not a process that the NADH and FADH2s are going to hold on to electrons for forever. It is kind of like thinking about there's a final destination where the electrons are going to be dropped off at, and the electron carriers are just getting the electrons to that point. They are the transportation to get the electrons there, because the goal is not to be on the transport, the goal is to get to the destination. But again, this is only for a short period of time that the electrons are going to be held by the electron carriers. The end goal of those electron carriers getting the electrons to their destination is to make ATP. That is the end-all be-all energy goal. So let's talk about what ATP is. You don't have to remember ATP as adenosine triphosphate, but I'd almost recommend remembering it only because it tells you exactly what ATP is made up of, and that's the important thing. So we have our nitrogen base, we have a 5-carbon sugar, but the most important part, and I want you to highlight this one, is the 3 phosphate groups. So these 3 phosphate groups are all going to have a negative charge. Each group of phosphate has a negative charge. And when you have molecules that have the same charge, all of them being negative, then those molecules don't want to stick together. They are going to try to repel each other. They are going to try to get some distance. So the reason that they are stuck together is because of a high energy bond that keeps them stuck together, even though they're trying to push and get away from each other. That's where the energy is going to come from when one of these phosphate groups pops off or breaks off. So that's kind of the big point here with this phosphate group, these 3 phosphate groups, is that the energy is going to come from one of those phosphate groups popping away from the other 2, which turns ATP, adenosine triphosphate, into ADP, which is adenosine diphosphate. So let's talk about that. When ATP loses a phosphate group, it becomes ADP. Now, this is a process called dephosphorylation, so the removal of a phosphate group. But because we don't want to just throw that ADP away, we are going to add a phosphate group, which is called phosphorylation, and it turns that ADP back into ATP. So we go in this really neat little kind of energy cycle, if you want to think of it that way, where we go from ATP to ADP, back to ATP, then to ADP over and over. It's kind of like charging your phone. You wouldn't throw your whole phone out once it lost its battery. You would just add energy to the phone by plugging it in with your charger to charge it back up. That is what we're doing with the ADP. Just because ATP doesn't have any energy doesn't mean it's useless. We're going to charge it back up, get it back to ATP. All right, we're getting in a little bit deeper to some of those terms we were talking about on the last slide. One that we find the most important right now is going to be phosphorylation. Remember that this is going to be the addition of a phosphate group in some capacity. So we want to discuss this when it comes to making ATP. There are three types of phosphorylation. The first is substrate level phosphorylation, and I want you to think of this as being like small potatoes. We are not making that much energy at a time in the form of ATP. It's just a small amount of energy being produced, as opposed to the second type of phosphorylation, which is oxidative phosphorylation, where we are going to speed up the process and produce a lot of energy in a short period of time. We're going to see this later in those metabolic steps to create energy. So those are the two most important ones. I'd put a star next to number 1 and number 2. The third one is photophosphorylation, and this is what happens when photosynthesis occurs. So that is that conversion of light energy into some form of chemical energy like ATP. This is the only time we're going to talk about that. It is just one of the three types of phosphorylation, but it's not even one we're going to discuss from this point on. We'll never talk about it again, but do know that it is a third type of phosphorylation, but number 1 and number 2 are the important ones as we move through these steps coming up. All right, we are jumping into that big process that we need to go through. And this is going to be carbohydrate catabolism. Remember that catabolism is going to be the breaking down of a larger molecule into smaller molecules in order to get energy. For this purpose, we're going to talk about the breakdown of glucose. So we have one glucose molecule at this starting point, and we're going to break that glucose molecule down into smaller parts. And this process is initially, the first one that we're talking about, is going to be a type of respiration. So there are two types of catabolic pathways for glucose, and that is respiration and fermentation. We're going to start with respiration, and then we're going to have to talk about the two types of respiration, which is aerobic, meaning it needs oxygen, and anaerobic, meaning it will be anything but oxygen. Okay, so from that point, we will talk about fermentation at the very end, but for the most part, guys, the big important couple of steps is going to be this respiration for aerobic and anaerobic respiration. So let's jump in. As kind of an outline, here are the 4 steps listed out. And this is going to be the same for aerobic and anaerobic respiration. Until we get to that last step, anaerobic respiration looks pretty similar to aerobic respiration. All first 3 steps and even most of the 4th step is pretty similar, but we'll talk about that difference in a bit. When it comes to what you need to know is what is the starting product that we are using for each step, and then what is the outcome of that step. So I'm going to kind of shorten this into what is the in and what is the out for every step. And then you also need to know what type of phosphorylation, if any, is happening during that step. So those are the 3 important things that you need to know for all 4 of these steps of respiration. Starting with step number 1, this is going to be glycolysis. I think it's easier to know what words mean when we break them down. So what glycolysis is saying is we are splitting sugars. We are breaking sugars apart. And that is exactly what's going to be happening here in step 1. So we are taking that glucose molecule and through a series of a ton of little steps that you don't need to know, we are going to break glucose into 2 molecules. They are the same molecule, but there's 2 of them. That is a pyruvate, or sometimes called pyruvic acid. But do know that there are 2 of them. We are also going to produce a little bit of energy on this step, 2 ATP molecules, and then 2 electron carriers, 2 NADH. So there are 3 products that we are producing through glucose, breaking apart, and we need to take into account all of those things. The other thing that we need to make sure we know is that this is going to be a substrate level phosphorylation step. The reason that it is a substrate level phosphorylation is because we have that production of a little bit of energy. Remember that substrate level is going to be small potatoes, while oxidative phosphorylation is like the big amount of energy. So we are producing a very small amount of ATP here, so this is substrate level. You should put a massive star or highlight this slide. It is going to be such a game changer and saver when it comes to studying. You should memorize this. This is the glycolysis net reaction. We have on the left part of that arrow the N. So that is the starting product. So it's that big glucose molecule. After going through glycolysis, we have 3 products, the out, that we need to memorize. We have 2 pyruvates, or pyruvic acid, same thing, we're just saying the same thing, and 2 ATP, which is what makes glycolysis a substrate level phosphorylation, that little bit of energy being produced. We also have 2 NADH molecules. Remember that those are our electron carriers. At this point in time, we're going to set that NADH over to the side, and we're going to use it in that 4th step, that 4th and final step. So these are our ins, glucose, and our outs, 2 pyruvic acids, 2 ATP, and 2 NADH that we're going to save for later. This leads us into step number 2, decarboxylation, or the creation of acetyl-CoA. It means the same thing. Now, what's important to make a very quick note of is that not all text and not all education materials might include this as a step. We are 100% calling this step number 2, but if you study from other materials, it might not include this as a step. Just know, I'm telling you right now, this is step 2 for us. That is what we are going to go with. Now, what's going to happen is those 2 pyruvates, those pyruvic acids that we produced from glycolysis are going to be the N, that starting molecules that we want for the creation of acetyl-CoA. So, what's going to happen is another couple of little mini steps. You don't have to memorize them at all. Just know the start is the 2 pyruvates or pyruvic acids. From there, the end part of decarboxylation is going to be 2 acetyl-CoAs, as well as some other things. A little bit of waste, we'll talk about it. Now, because we have no ATP being produced, even in a small amount, on this step, that means no phosphorylation is happening. So make sure you know that. Step 2, we start with the pyruvic acid molecules, and the end product is going to be a little bit of waste. We're also going to produce a few electron carriers, as well as there is no ATP produced, and we are producing acetyl-CoA. So no phosphorylation because no energy has been made at all, at least in this step. Now, again, this slide is going to be a lifesaver. I want you to remember this reaction. So remember, this is step 2, decarboxylation, or the creation of acetyl-CoA. The N is 2 pyruvic acids, or 2 pyruvates. The out, the product, is 2 acetyl-CoA, as well as a little bit of waste, which is 2 carbon dioxides. And then we also have our electron carriers being produced as well, and that's 2 NADH. So again, kind of keep in mind, the electron carriers that we produce in these first couple steps are going to be used in that final step, step 4. So these guys are going to wait until the end, but they have been produced. those two acetyl-CoAs are going to go into the next cycle, or the next step, which is going to be a cycle called the Krebs cycle. And then that carbon dioxide is going to be a waste product, and also because carbon dioxide is the waste of this process of producing energy. Fun fact, it's why we exhale carbon dioxide, because we produce it as a waste of making energy in our cells. Super cool, right? On to step 3, the Krebs cycle, sometimes called the citric acid cycle. Just remember one of them. If you know both, cool. I love that for you. If you want to memorize just one of them, that is perfectly fine too. So this is step 3. We are starting with the end product of decarboxylation, which was those two acetyl-CoAs. From here, again, a couple of mini steps that we don't really care about, but in the end, that is what we care about. We are going to produce a little bit of ATP in the Krebs cycle, just like we did with glycolysis. That means that the Krebs cycle is going to be substrate level phosphorylation. Just a little bit of ATP is produced because we're squeezing that last little bit of energy out of the acetyl-CoAs. Because remember, we have broken down glucose into pyruvic acid, and then that pyruvic acid we have broken down into acetyl-CoA, and we're going to squeeze out a little bit of energy before we move on to that final step. Now, once again, I would memorize this. This is that net reaction of the Krebs cycle. We have those initial two acetyl-CoAs that we start with at the start of the Krebs cycle. That is our in. The out of the Krebs cycle is 6 electron carriers in the form of NADH, 2 more electron carriers in the form of FADH2, a little bit of energy, 2 ATP, and more waste for carbon dioxides. Now, it's important to note here that that little bit of energy again means that we have substrate level phosphorylation happening. We have more electron carriers that we are going to again add to the end step. So they're going to move into that step 4. And then we have that little bit of waste. Again, carbon dioxide, that is why we breathe it out as a waste product. So let's review what we have covered thus far. The first step was glycolysis, then we had decarboxylation and the Krebs cycle. In glycolysis, we start with glucose. That is the in. The out of glycolysis is going to be 2 pyruvic acids, 2 ATP, and 2 NADH. Remember that the pyruvic acid is going to be what we start with for decarboxylation, that second step. The 2 ATP indicate that for glycolysis, that is substrate level phosphorylation, and those 2 electron carriers are going to wait until that 4th step, so we're going to set those to the side. For decarboxylation, again, we start with the 2 pyruvic acids that we produced in glycolysis. From there, the out is 2 acetyl-CoAs, which is going to go into the Krebs cycle. We have 2 carbon dioxides, which is a waste product that is just going to be exhaled. And then 2 NADHs, also electron carriers, that is going to be pushed to the end of our entire process here into step 4. In the Krebs cycle, we have 2 acetyl-CoAs from decarboxylation, where the end product is going to be the 6 NADHs, 2 FADH2s, and 2 ATP, and a little bit of waste for carbon dioxides. So those electron carriers, NADH and FADH2, are going to wait until that final 4th step. Now at this point, guys, I want you to make sure that you know that when it comes to the electron carriers, it's going to be additive. So at this point, we have 10 NADHs and 2 FADH2s, as well as we've produced 4 ATPs through this process. Again, very small amount of ATP being produced, and in the two steps that have produced ATP, glycolysis and the Krebs cycle. That is substrate level phosphorylation. Remember that decarboxylation did not produce any ATP, so there's no phosphorylation happening there. Now, let's move into that fourth and final step of respiration. All right, we are moving into the fourth and final step of respiration, which is the electron transport chain, or the ETC. At this point, all of those electron carriers are going to come into play. So we have the 10 NADHs and the 2 FADH2s. These guys are going to bring electrons into this chain, which is going to be a chain of enzymes. What's going to happen is that this chain of enzymes is going to pass the electrons that the electron carriers have provided from one to the other. What this is going to cause is hydrogen ions that are positively charged to move across the plasma membrane and be outside of the cell. On the inside of the plasma membrane, those electrons are being passed from one enzyme to the next, which is going to end up causing a negative charge through the plasma membrane, as opposed to the outside of the cell, which has those positively charged hydrogen ions. Now, when we have a positive and negative charge, they are going to want to stick together. That's kind of an important thing to remember. What we are creating with those hydrogen ions being outside the membrane is an electrochemical gradient. There is a high amount of positivity on the outside of that plasma membrane and a negative charge on the inside. The other thing that's happening is that there's a lot of hydrogen ions on the outside, and on the inside there is not that many. So those hydrogen ions are going to want to come back into the cell, but they're going to need a way to do that, and we're going to talk about that in a second. Let's first talk about what's happening with the electrons in that enzyme chain. In the enzyme chain, those electrons are being passed from one enzyme to another until they reach the end of the chain, and then there needs to be something there, a molecule, to catch that electron. The molecule for aerobic respiration that catches that electron, known as the final electron acceptor, is going to be oxygen. Aerobic respiration, that final electron acceptor, is going to be oxygen forever and always. What this is going to produce is water. It's essentially a byproduct. Now, it's important to note that when aerobic respiration happens and oxygen is that final acceptor, what we have is oxidative phosphorylation, because this is going to end up producing a lot of energy. But we also need to still talk about what's happening with those hydrogen ions and how they get back into the cell from that area of high concentration to inside the cell, low concentration. So I'm going to show you an image first, and then we're going to talk about what's happening. All right, we have the membrane of the cell, and those big circles, those blue circles, are going to be those enzymes that are passing electrons through the chain. We have our electron carriers providing those electrons into the chain as well. And then we have at the end of the chain oxygen waiting to catch the electron and then produce water. Again, this is making that gradient where those hydrogen ions are able to move across the plasma membrane and out of the cell. But then we have that high charge, that positive charge, as well as a larger amount of hydrogen ions on the outside of the cell that are wanting to get back in. Because remember diffusion, we are going from an area of high concentration, so a lot of hydrogen ions, to an area of low concentration, which is the inside of the cell that is going to have a low concentration of hydrogen ions. What is going to allow those hydrogen ions to come back into the cell is going to be something called ATP synthase. That is going to be that molecule you can see in red on the far right. It creates essentially a doorway that allows the hydrogen ions back into the cell. But something has to happen in order for those hydrogen ions to be allowed back in. Let's talk about it. I apologize, I did change the order of the next two slides. So if you're looking at this and you're looking at your notes and saying, where are we? I have just moved the next slide up one. So they're still very close to each other. It's just I've moved this slide very quickly. So with ATP synthase, what is happening is we are making, because remember synthesizing means to make, we are making ATP. So that doorway that ATP synthase allows hydrogen ions to move back into the cell. As the hydrogen ions go through that channel, they have to pay a toll, essentially, to ATP synthase in order to get back into the cell. It's kind of like going through a turnstile on a subway, right? You have to pay to get through the turnstile. What the hydrogen ions are paying is that they have to convert ADP into ATP. They have to add that phosphate group back to the ADP so that it has been phosphorylated. Because the end result is we want ATP. So for those hydrogen ions to pass through, they are making ATP molecules. Now, kind of going a little bit backwards so we can move forwards, when an NADH is giving electrons to the enzyme chain, once it does that, it produces enough hydrogen ions to make 3 ATPs when those hydrogen ions move back into the cell through ATP synthase, as opposed to FADH2, which for every FADH2 molecule providing electrons into the transport chain, it is able to produce 2 ATPs. So our electron carriers are not equal, and it's important to know how many ATP molecules are going to be produced per electron carrier and which type of electron carrier we're talking about. So where is all of this occurring? That is going to be the membrane of most cells, but it's kind of important to note that eukaryotes, this is going to happen in the mitochondria, specifically the inner membrane of the mitochondria found in our cells. This is just for eukaryotes, because remember that prokaryotes, things like bacteria, don't have mitochondria, so they don't have that little powerhouse to make the energy that their cells need. So for prokaryotes, where this is going to happen is in their cell membrane. So it's kind of interesting to look at mitochondria, because remember the theory of endosymbiosis, that mitochondria was just essentially a prokaryote that was consumed by another prokaryote, and now mitochondria makes energy for our cells. But we have a lot of mitochondria, which is kind of a neat fact in why eukaryotes were able to become larger and multicellular. It's because we had these little batteries producing energy in the cell, as opposed to the cell itself, like a prokaryote, a bacteria, having to make it for themselves. So instead of having a symbiotic relationship, prokaryotes have to do all their own work and produce their energy, which is maybe a reason why they stay single-celled and very small. All right, our slides should be back on track now. So This is the electron transport chain and a breakdown of some numbers that we need to go through. The first is how many NADH did we have at the end of step 3? And the answer is 10, because from step 1 we had 2 NADHs, from step 2 we had 2 NADHs, and from step 3 we had 6, giving us a total of 10 NADHs, so those electron carriers from all the previous steps before the electron transport chain. For each NADH molecule, we are going to get 3 ATP. So we have 10 originally. We're going to multiply that by 3. That gives us a total of 30 ATP from NADH alone. From there, we need to talk about our FADH2 molecules. We have, in total, 2 of these guys. Just from step 3, we only produced 2. But for each 2, we get 2 ATP. So 2 times 2, we get 4 ATP in total. So if we add that together, we should get a nice solid 34 ATP made through oxidative phosphorylation in the electron transport chain. Again, I want you to think of substrate level as being a small amount of energy being produced, and that is what we produced in steps 1 and 3. And in this last step, step 4, the electron transport chain, we are producing a massive amount of energy, 34 ATP all at once, which is a very good amount. But it's not actually our total amount of energy. Because remember, we produced a little bit of ATP in steps 1 and 3. So in glycolysis, we produced 2 ATP, and in the Krebs cycle, we produced another 2 ATP, where in this last step, we produced 34. That is going to give us a total of 38 ATP molecules. So for respiration, for aerobic respiration, you need to know that number. That is 38 ATP. And that's if everything goes perfectly, which it does not always go perfectly. We don't always get 38 ATP through aerobic respiration. But for the purpose and simplicity of this class, I want you to imagine it is always perfect. We always produce 38 ATP through the process of aerobic respiration. That now leads us into anaerobic respiration. It's the other kind of respiration. We're still going to produce quite a bit of energy with this process, but that final electron acceptor is not going to be oxygen. Very, very important thing to make a note of. Highlight that piece of information. The final electron acceptor is not going to be oxygen. It's going to be some other inorganic molecule, like sulfur or something else, okay? It just depends on the species that we're talking about. It will be a different acceptor at the end process. So that electron transport chain will still happen. It's just modified because it can't be oxygen because it's anaerobic, meaning no oxygen. Now, comparatively to aerobic respiration, this isn't as good because you will not produce as much ATP. It could be anywhere between 4 ATP and 37. But what I want you to remember is that the yield of ATP, that final amount, is always going to be less than 38 ATP. So, gentle reminder, oxygen is the best electron acceptor because it will produce the most ATP. But anaerobic is still good, but it will not produce as much ATP, always less than 38. Now, it's not always glucose and carbs that can be broken down for energy. Other macromolecules can be broken down into the respiration cycle, but they're going to start at different points of the respiration cycle. And we're not always going to yield as much ATP in that process. So carbs and glucose will give us the most energy, kind of more bang for your buck, if you want to think of it that way. It's just other macromolecules are going to produce energy less than carbs, but they're still good. All right, so that was all of respiration. Let's talk about fermentation. Fermentation is only going to use one of the steps that we talked about with respiration, and that is glycolysis. Remember that in glycolysis, we produce 2 ATP and some electron carriers. But what's important to remember here is that there's only 2 ATP being produced if that is the metabolic pathway that a cell chooses. And I say choose very lightly because if you're only producing 2 ATP, why would you ever choose fermentation? Well, the answer is there's probably no other option for the cell. So this is usually a last resort for a cell to produce that last little bit of energy that it can. So for us, I want you to imagine that fermentation would have happened in humans, because it does happen in our cells, it can, but it's usually going to be like you're running for your life from a bear or a tiger or whatever. Now, for our cells, this is not a good thing. You don't want to be using fermentation as that production of energy. We want aerobic respiration. That is the best way to produce energy. But if your cells are essentially at their limit, they cannot go through aerobic respiration because maybe you're huffing and puffing from running from that predator. You are low on oxygen, so that final electron acceptor is limited in your system. Your cells will switch to fermentation to try to help you survive. Now, again, it is a last result for our cells. And the product of fermentation within the cell is usually not always a good product, like a byproduct, if you want to think of it that way. For our cells, it's lactic acid. That is going to be what builds up for us when you feel that burn in your muscles or start having cramps. So we're not typically going to be running for our lives from predators anymore. However, you might be running your butt off on the treadmill or doing the stair climber or, climbing a wall for whatever reason, parkour, whatever. In that process of exercising, maybe you're pushing yourself really hard, pushing yourself to that point that your cells need to switch to fermentation, and that is what causes that buildup of lactic acid in your muscles. For other organisms, they are going to have other types of byproducts due to fermentation. Again, this is usually a last resort type of metabolic pathway. So those end products can be anything from lactic acid to ethanol, to other types of alcohol. We'll talk about that here on the next slide. But other cells, like bacteria, if they have to switch to fermentation to produce energy, then it is likely that they are going to have some type of byproduct such as alcohol, acids, and gases. Some microbe fermentation byproducts include alcoholic beverages, some solvents, other organic acids, as well as vitamins, antibiotics, and hormones. Again, fermentation for us isn't going to produce as cool of a product because lactic acid is just going to make your muscles burn. But for these microbes, bacteria, fermentation is not going to be an end result of the cell being unharmed. So it can still be kind of detrimental to the cells, but when it comes to kind of large scale industry production of fermentation products, they are just microbes, so it kind of makes you feel less bad about kind of putting the cells through the ringer in order to get alcohol or vitamins or hormones or antibiotics, etc. I would put a big ol' star next to this slide. This is a nice overview of those three main catabolic reactions that we have just talked about. The first two are our two types of respiration, aerobic and anaerobic. These guys are going to be important because they produce a metric crap ton of ATP. Remember, for aerobic respiration, we are starting with glucose and going through those 4 steps to get to that end product of 38 ATP. And oxygen is going to be that final electron acceptor. Very, very important. For that second type of respiration, anaerobic respiration, we go through all the same steps as aerobic respiration, but that final electron acceptor is going to be something other than oxygen, and we will always produce less than 38 ATP. In fermentation, this is kind of a last result if we can't do aerobic or anaerobic respiration. Oxygen is not required because the only thing that's happening is glycolysis, that first step of respiration, and that's it. There are only going to be 2 ATP produced, and also some type of byproduct is usually going to happen as a result of fermentation. Once again, a very nice overview of what is occurring and also the end products of these 3 types of catabolic reactions. Make sure you know it for the test. All right, so up till now, we have talked about primarily the catabolic pathways to get energy within a cell. So remember that catabolic means that we are going to take some molecule and then break it apart to get energy, while anabolic is going to be kind of the opposite of that. We're going to use energy to build a molecule. So let's very quickly, very simply look at that process. There are, in fact, a couple million ways to look at these anabolic pathways. We are going to massively simplify it and just look at the production of macromolecules, things like proteins and lipids and nucleic acids. Now, we have already talked about the different building blocks of these macromolecules. So remember that for proteins, it's amino acids. That is what builds a protein. The lipids are going to be fatty acids and glycerol, where nucleic acids are going to be made up of nucleotides. For anabolic pathways, it's going to be this process of what are we building? So let's just look at these macromolecules as our example. We are building these molecules from their little building blocks. So again, we are going to have to use energy to build a molecule. That is anabolic. So what might a microbe need to build? For bacteria, what they're going to be building is all of the components of that cell. And the reason that they do this is once they hit double of everything inside the cell, it's going to trigger something in a bacteria or prokaryotes in order to go through binary fission. So once these products have been synthesized or made, the cell is going to feel the need, I suppose is a good way to say it, to make a new cell. So they go through binary fission, make a new cell. So that is one of the ways that our microbes are going to use that anabolic pathway, is to build all of the structures inside of that cell. And with that, guys, that is the end of chapter 7. There will be a review posted in our Brightspace that is going to go through the main points of Chapter 7, because I know that this was a really heavy chapter, and I'm so glad that you stuck it out and finished it up. So keep reviewing. If you have any questions, don't hesitate to ask or email me and work on that review. So remember, make good choices, study up. The exam is right around the corner. We only have one more chapter left of this unit, and then we are going to have an exam.